The first is a really important project, particularly in the context of the recent developments in the PWS field. So one of the challenges that we have in clinical trials in PWS is really showing that a drug is effective. And that has been a challenge for pretty much every drug that has come into the PWS field. And in some cases, we as parents, we can see that a drug is having an effect. And I'm thinking of LV-101, the levo drug in this case, which recently had that FDA advisory committee meeting that was unsuccessful. So we can see the differences, but the FDA has to be able to quantify that and see it and be convinced that there is a change in hunger in order to get the approval. And part of the issue that we currently have is the reporter-based systems, the questionnaires that we use. They're good. They capture many aspects of hyperphagia and PWS, but sometimes they're just not sensitive enough, likely to detect some changes that might otherwise be meaningful. So we need to build up that complement of endpoints and assessments that we can use in clinical trials. And so Dr. Gallagher is going to be building off some work that has been done at Vanderbilt University, kind of taking it to the next step, looking at eye tracking and attentional bias as a potential biomarker and endpoint in clinical trials. So this study is going to look at eye tracking as a biomarker for altered satiety in PWS using a computer-based system. This is a very experienced researcher. Her initial focus has been in autism, but she's also been doing some work recently in PWS, and she also has experience in developing endpoints for autism clinical trials. So she brings that experience to the study. And obviously, this is an area that we need to be bolstering up so that drugs that have an effect, but maybe not a meaningful, but not necessarily, dramatic across the board effect, can still demonstrate effectiveness in clinical trials so that we can reach approval. So the long-term goal is to create a robust, objective biomarker of hunger and satiety that can be used to monitor treatment effectiveness for hyperphagia in PWS.